Okay, thank you, Olivier. So picking right up from where Anna left off, assuming that you've now packaged your virus into a high titer virus and you've established what the titer of that virus is, you're now ready to actually start the CRISPR screen itself. Just kind of an overview. Initially I had 4 points, but I added one more, that if you're going to use a version of the virus that doesn't carry Cas9 with it, so this would be like the Gecko version 2, the Gecko Lentigai Puro. You need to establish that you have Cas9 already in your cells. As Olivier and Anna have mentioned, you have to remember that coverage is important to maintain throughout the entire experiment. Remembering that these are competitive growth assays will help us kind of think about how we analyze our data. And then based on what you want to ultimately discover, that will change many of the parameters in your experiment. So it's important to keep that in mind from the beginning. I'll just say that we're just going to talk about a few variations. If you have other questions, obviously these are just a couple of examples of potential screens, but there are many other variations. If you have more questions, please hit me up. So, in order for Cas9 to cut the genome, it needs to be present. And unfortunately, Cas9 expression can be quite transient over time. And so what this means is that just because a cell is labeled as Cas9 expression, don't take that for granted. Your colleague that gave you that cell line might have validated that Cas9 was expressed at a high level, but after 4 weeks of passage, that Cas9 expression might have decayed. And that could be true even if the cells have undergone clonal selection. So it's important to validate that. You could do it by qPCR. We like to use a Western, and we actually use an electropherogram, which is similar to a Western. It's a proteomic-based approach, but it's a little bit faster. The gold standard really would be to actually do a next-generation sequencing experiment where you PCR up the target of your guide, and you can count all the reads and identify how many have been cut. We're using a protocol that's adapted from the Mali Labs protocol to do that, and you can use this software called CRISPR, which is free and on the web to analyze that data once you have it. In our experience, we found that using a lentiviral to randomly integrate Cas9 into the genome gets you as much Cas9 and as stable Cas9 expression as cloning Cas9 into a safe harbor locus like AAVS, although that result is likely cell line dependent. But at least in our experience, we've done just as well with random integration with Cas9 with lenti. Okay, so again, this is somewhat redundant, but it's important to know that this is a competitive growth experiment. At the end of the day, you're going to get a next generation sequencing result. If you see more copies of your guide RNA, that means that guide RNA, edited the genome, and that resulted in a growth advantage. If you have a decreased number of reads for that guide RNA, that means that guide RNA, when it cut the genome, led to a growth disadvantage. Because your total number of reads is capped, this is a compositional data set, and like microbiome research, whenever you have an arbitrary cap on the number of reads, you can't make comments about the absolute growth effects, but only the relative growth effects. The other thing is that this is an evolutionary-based experiment. We're editing the genome, and we're seeing what the fitness effect is, and evolution takes time. So I'll talk about time in days, but really the best way to think about how long your experiment should go should be relative to the doubling time of your cell line. So if you have a slow-growing cell line that takes 4 days to double, you're going to need to run the experiment longer to get the same results. And because of that, though, in our experiment, and also because of the number of cells that are involved, we find it's best to do these screens, if possible, in cells that grow relatively rapidly. We've screened about 7 cell lines in our lab, and the only 2 that have had poor results were some ovarian cancer cell lines that were very difficult to maintain in exponential growth. So again, we'll harp on this again because this is, I think, the single most important idea in a pooled CRISPR screen is that you have to maintain coverage of your library at every single step. Using the Gecko library as an example with 120,000 elements, coverage is how many copies you have on average. Obviously, this is not, it will be a Gaussian distribution. 1,000 elements is represented 300 times, and that would give you would need 36 million cells to do that. So a large number of cells. How much coverage you need is going to depend on what you're trying to find. If you're looking for things that deplete, you're going to need a higher coverage. If you're looking for things that enrich, you could get by potentially with the lower coverage. And so again, talking about coverage, the thing is, if your experiment bottlenecks at any level, if it bottlenecked at the plasmid level, if it bottlenecked at the virus level, if it bottlenecks at the cell level, you can't get those lost reads back. And the thing is that even if you sequence more at the end, if you've lost, say, half of the elements in your library, if you double your sequencing depth, you're just going to get more reads on the half of the library that you have because the part that's lost is gone. So sequencing your library, if you have a nice distribution like this, you get a nice Gaussian where your population has not been bottlenecked. If you sequence this more, this average will move to the right, and that would give you more power to detect things that drop out. But if you bottleneck your library, if you sequence this more, this peak is just going to get, this peak will move to the right, but these genes that are present at 0 amount of the time won't magically reappear at the end. So it's important that you don't bottleneck your library. Okay. So at the beginning, you need to ask yourself, do I care about what genes are dropping out that cause a fitness defect? Do I care about what genes are enriching, or do I care about both? For example, if you're doing like a drug resistance screen and I want to know when I mutate this gene, because most of these mutations will be loss of function, does that cause resistance? You'll need less coverage because the things that you care about are going to be present at higher sequencing reads. Whereas if you care about what sensitizes to your perturbation, that's going to be dropping out. And so if you don't have a very high average copy number, you won't be able to detect with statistical significance something that dropped out. If you need to know, if you only care about the outliers, for example, if you're doing a single knockout screen, you probably don't care if gene X has a 1% growth effect or a 2% growth effect or a 5% growth effect. You only care about the things at the extremes. In that case, you don't need to know with nearly as much precision the single gene. In that case, you can, again, get away with less coverage and probably only doing two time points. If you do need to know the fitness of every gene precisely, for example, to look for genetic interactions, which is what I'm primarily interested in, we use multiple time point scaling and fit regression, and that gets you a much more accurate estimation of fitness for things that don't have a large effect. And then the list of, do you already have prior knowledge about what genes are important, or do you want a genome-wide screen? This goes all the way back to the idea of library design. I won't talk a lot about library design now because I think most of you will probably use a commercially available library. But if you have questions about designing and synthesizing a custom library, this is what I've spent a lot of the last 6 months of my life doing, so I'm happy to talk to you about that offline. Okay. Really, as has been said by many, the elegance of any experiment is the positive and the negative controls. Because next generation sequencing is a somewhat noisy readout, in order to have confidence in your data, having high confidence positive and negative controls is really critical. There is a lot of inherent randomness with the transduction of the virus into the cells. And so it's important to, when you get these fitness measures back, to make sure to convince yourself that this is not just randomness. And so the controls will depend on the experimental design of your particular screen. One thing we like to do is we like to run a small screen with no Cas9 expression, because then you should have no selection. And that will also give you an estimate of how big the signal that you're seeing, how big is that relative to a randomized control. It's great if you happen to know specific genes that should have a large fitness effect as a positive control. That's also very helpful. For a lot of cell lines, that would be genes like P53, or if you have a cell line that it's an EGF mutant or KRAS mutant, including, you might think, I don't want to include KRAS because it's not interesting. I know the cell's dependent on KRAS. In fact, absolutely you should include that, because having that positive control will be very helpful, and that will also allow you to scale the magnitude of the effect of your new genes to the known biology. So here's an example of our Cas9 null control. So what you're looking at here is 2 independent transductions, and looking at the correlation between the replicates. F sub G stands for the fitness effect of knocking out the single gene. So you can see on the left here that we have very nice, this is like a 0.95 correlation between our 2 replicates. Gives us a lot of confidence in the data. We see that the genes that are having a largely strongly negative fitness effect are genes that we know are important to the cell, things that involve growth factor pathways and DNA damage checkpoints. and cell growth. And then, but giving us even more confidence is we see that, this is about what we would get just randomly with noise when we don't have Cas9. So we know that, maybe these genes are also significant, but the things up here are clearly not. And so having that negative control really helps you scale the results of your real data. Okay. Other important caveat to know is that the guide RNA, assuming you have a well-designed guide RNA, it should only target one area of the genome. However, I'm a cancer biologist, and I feel like many of you are also cancer biologists. Cancer has a notoriously deranged genome, and if your guide targets an area that is genomically amplified, like here, that means that Cas9, your guide is going to bring Cas9, and it's going to cut at every one of those amplified copies. And if you have more than 2 or 3 breaks in the DNA introduced by Cas9, that's going to cause the cell to grow slowly, independent of the gene-specific effects. And so what you can see here, this was shown first by the Bill Hahn group, that they're targeting even actually intergenic regions that are copy number amplified, and they're scoring as negative hits. And so they show this is specific to a CRISPR and not seen with RNAi. So it's important to know the copy number data for your cell line, because if the gene is heavily copy number amplified and you send a guide there, it's going to look like it's going to drop out, even if that gene isn't actually important. Interestingly, and thankfully, the Hahn group has also come up with a computational fix for this. So you can see this paper just came out about a month ago in Nature Genetics. This is actually a great resource. One, for this series algorithm, which corrects for the copy number effect. It's also great because they have data on single gene knockout for, I think, 343 cell lines with the genome-wide library. So a lot of the data that you want may already be there. But you can see here that with uncorrected data, as copy number increases, you can see the fitness effect decreases, but that's largely normalized away with this algorithm that they call series. And basically this is because the normal cell response to lots of DNA damage is to invoke DNA damage repair, and that's going to put you into a cell cycle block, and therefore your cell is less proliferative. Okay. All right. So the 2 most common experimental designs are we want to find what genes are essential to the cell line, and this is essentially the experiment that Hahn has already run in those 340 cell lines. Or the next most common experiment would be, I have a drug of interest, I want to identify either things that sensitize to that drug or things that cause resistance. And so in this case, you would run your pooled screen, plus and minus your drug. And so you can either, if you do a drug screen, you can, whether you're more interested in looking for resistance or sensitivity will be important, because if you're looking for resistance, you need to have a dose that will not kill most of your cells, whereas if you're looking for sensitivity, if you're looking for sensitivity, you're going to need a dose that will not kill most of the cells and look for what is killed. If you're looking for resistance, you'll need a dose that will kill the majority of your cells, and then see what's left. Okay. So just kind of just the mechanics of how this works. If you use cells that double about every 48 hours, we like to run these experiments for a total of 28 days. So that's 14 cell doublings. We found that we get good selection with that. You could try shorter, but I think if you have the ability to run the experiment longer, it's better to run it longer. The very first day, you will plate your cells. We call day 0, the day we add the virus. Again, as Anna mentioned, it's important that you've established the titer, because you want to make sure that that each cell only picks up 1 virus. Because our readout is going to be sequencing at the end, we have no way to tell if that guide was in the same cell as another guide, and so that's why we have to maintain only 1 guide per cell. The way we do that is we have an excess of cells to virus, so that we expect that when we start our puromycin selection, we're going to kill about 60% of the cells. So if you get to this point and you don't see that about 60% of the cells are dying, then you've either your pure selection is not working, and you haven't established an appropriate puromycin killing curve, or you've put too much virus in and you've pushed the MOI beyond 0.3, and that's really convoluted your data. You're not going to be able to analyze the data. Freezing at the initial sample is very important because ultimately we want to know what enriches or depletes, and so the best comparison, in my opinion, is the time zero sample. You could argue that you could use just the representation of the library in the plasmid pool as the initial sample, but because despite having a high titer virus, the process of integrating the library into the cells is somewhat random. You'll get a lot less variance if you use the initial post-transduction DNA as your, which is all the virus that made it into this replicate as your initial T0 control versus using the plasmid pool. Okay. And so again, just a little bit of math. So again, we have to so it's important to maintain coverage in the frozen part because we're going to extract that DNA and sequence it, and it's important to maintain coverage moving forward. So that means that we can't, if we want to do 300x, we can't just have 300x and freeze it. We have to have 300x moving forward and 300x frozen. So if we have, if we want 100x coverage and our library size is 120,000 elements, that is 12 million cells to preserve that complexity. So that would mean that's 12 million cells to freeze and 12 million cells to carry forward per replicate. So we would need 24 million cells in our dish. That is a lot of cells. To do that, we normally use bioassay plates, which have a surface area of approximately 500 square centimeters. It's like a 22 by 22 centimeter square dish. And with cells that pack into high density, you can achieve that. So again, it's another reason why if you're doing this with a very slow-growing cell line, it will be very difficult. So if we're just looking for essential screens, so this is an experiment with no drug, we'll do that. We'll add our puromycin. We keep the puromycin in throughout the experiment. You'll plate back your 12 million cells. Because you're putting 12 million cells into a dish, they're going to get confluent probably in about 2 to 3 days. We need to keep the cells in exponential growth. So after 2 to 3 days, we'll need to lift the cells, count them, and patch them again. Every time, we generally save all these cells, even though we won't actually go and sequence most of them, but it's better to have them in case, for example, this PCR would fail. You've got, maybe day 14, the sample got thawed or somehow lost. You would have, the day 12 or day 9 sample frozen. So again, at every point here, you have to passage at least 12 million cells forward. As the experiment goes on later, that should be less of a problem because, by day 6, your puromycin selection should be complete. And you should get, depending on your cell line, you should be able to get into a nice pattern where you know about how many cells to plate to reach confluency 3 days later, and then you can carry this forward. We'd like to do it for about 14 doublings. And so we do that. You can plot. So because you're removing about half to 2/3 of the cells at every passage, the cumulative cell count is not going to be exponential. It should be linear. It's important to record your cell counts at every passage so you can see if there are any issues. You also want to make sure that the cells have a nice constant doubling rate, which you can see here. So based on the number of cells that you plated and the number of cells you count, you can calculate a doubling for every interval harvest, and you should see a relatively constant doubling rate. If that's not going on, then that tells you that there's an issue with your cell culture. It's important we like to do a minimum of 2 replicates, so that's 2 independent transductions per experiment. That helps make up for the fact that the viral integration is random. And so doing this, we'd like to see about a 0.9 correlation in terms of single gene fitness between the replicates. So that's, a single essentiality screen, no drug. It's not that much different with the drug. The basic idea is that we can either apply a fairly heavy selection, which will kill most things and just kind of pick out the winners, or we can apply a relatively lower level of selection and then see what enriches. So here, obviously, it's important that you establish with confidence what the dose response curve of your drug is in your cell of interest. If we were going to do a positive screen, we're going to pick a dose that's going to be high enough to kill most of the cells. The initial part will be fine just because putting the mechanics of doing the initial transduction of the library into the cells is the same. But after we finish pure selection, we'll go into drug, and then we're going to compare this with drug to the without drug samples. The one thing of note here is that because if we're doing a positive selection screen and our drug dose is going to be significantly toxic to the cells, this will get passaged a lot less than this one just because the drug is clearly slowing down the growth of these cells. Negative screen, it's essentially the same. The only difference is that we're going to now pick a much lower dose of the drug. Because of that, we'll probably passage the with drug cells just about as frequently as the no drug, and then we'll compare the sequencing results of these at the end. Okay. And just in the last little minute, so most of the time, this is done just to look for viability. Do the cells grow faster or do the cells grow slower? Obviously, there are a lot of phenotypes that are important other than death. One interesting way to do that is you could use, if you have a fluorescent reporter for your either gene of interest or your phenotype of interest, you could fact sort into your GFP high or fluorescent high, fluorescent low, and sequence those two different populations. And that way you could, as long as you can separate the phenotype by a flow or other selective marker and sequence them separately, you could use the same basic idea to look for more complex phenotypes. And then finally, just a word, I'm primarily interested in genetic interactions, and so we've built a construct that delivers not one, but two guide RNAs, where we know both constructs that go into each cell. We use a multiple time course to estimate the single guide RNA fitness with higher accuracy, getting you kind of a much more accurate estimation of fitness, even for genes that don't have a large fitness effect. And with that, you can identify pairs where the double knockout effect is significantly different than what you would have expected from the combined single knockouts. Okay. So I went through that kind of quickly, so there's questions now, or otherwise you can grab me at the break.